Animal viruses. Most viruses that infect animals have a membranous envelope, viral glycoproteins on the envelope, bind to specific receptor molecules on the surface of a host cell. Some viral envelopes are formed from the host cell's plasma membrane as the viral capsids exit, while other viral membranes form from the host's nuclear envelope and are then replaced by an envelope made from Golgi apparatus. Bacteriophages aka phages. These have the most complex capsids found among viruses. They have an elongated capsid head that enclose their DNA. A tail piece made of protein acts to attach this guy to the host and inject their DNA. Capsid. The protein shell that encloses the viral genome. They are built from protein subunits called capsomeres. It can have various structures, depending on the type of virus. This can be rod-shaped, polyhedral, or more complex shapes. Capsule. This is a layer of protein or polysaccharide that covers many prokaryotes. Evolution of viruses. Viruses do not fit our definition of living organisms. Since viruses can reproduce only within cells, they probably evolved as bits of cellular nucleic acids, candidates for the source of viral genomes are plasmids, circular DNA in bacteria and yeasts, and transposons, small mobile DNA segments. Plasmids, transposons, and viruses are all mobile genetic elements. Mimivirus, a double-stranded DNA virus, is the largest virus yet discovered. Host range. This is the limited number of host cells that a virus can infect. This host specificity results from the evolution of recognition systems by the virus. Viruses usually identify host cells by a lock and key fit between viral surface proteins and specific receptor molecules on the outside of the cell. Lysogenic cycle. This process replicates the phage genome without destroying the host. The viral DNA is known as a prophage. Every time the host divides, it copies the phage DNA and passes the copies to the daughter cell. Lytic cycle. This term refers to the last stage of infection, during which the bacterium lyses, breaks open, and releases the phages that were produced within the cell. These new phages can then go and infect new cells. This is the reproductive cycle of a phage that culminates in the death of the host cell. This process produces new phages and digests the host's cell wall, releasing the progeny virus. peptide or glycan. These are contained within bacterial cell walls and consist of a network of sugar polymers, which are cross-linked by polypeptides. Prions. These are slow-acting, virtually indestructible infectious proteins that cause brain diseases in mammals. These propagate by converting normal proteins into their own version. Prophage. When the viral DNA is incorporated into the host cell's chromosome, the integrated viral DNA is called this. Provirus. What the viral DNA that has been integrated into the host genome is called this. In contrast to a prophage, remains in the host cell permanently. The host's RNA polymerase transcribes the proviral DNA into RNA molecules. The RNA molecules function both as mRNA for synthesis of viral proteins and as genomes for new virus particles released from the cell. Restriction enzymes. This is the defense mechanism that bacteria have against phages. This recognizes and cuts up phage DNA. Retrovirus. This is the use of reverse transcriptase to copy their RNA genome into DNA, instead of DNA to RNA, which is the normal process. HIV and other retroviruses are enveloped viruses that contain two identical molecules of singe-stranded RNA and two molecules of reverse transcriptase. Example: HIV is the retrovirus that causes AIDS. Temperate phages. This is what phages are called when they utilize both the lytic and lysogenic cycle. Vaccine. Harmless derivative of pathogenic microbes that stimulate the immune system to mount defense against the actual pathogen. These can prevent certain viral illnesses. Viral infections cannot be treated by antibiotics. Antiviral drugs can help to treat, though not cure, viral infections. Viral envelopes. These surround the capsids of influenza viruses and many other viruses found in animals. These are derived from the host cell's membrane, and contain a combination of viral and host cell molecules. Viral replication cycle. Viroids. Virulent phage. Viroids. These are circular RNA molecules that infect plants and disrupt their growth. Virulent phage. This is a phage that reproduces only by the lytic cycle. Virus. Virus.